Well, thanks everybody for joining the Brain Space Initiative this week. It's my pleasure to introduce Richard Betzel, who you like to go by Rick or Richard, or you don't care? Yeah.
Great. I've been called so many things. Yeah, whatever you want is good.
Okay, so be careful what you ask for, yeah. Anyway, thank you for joining us. So Rick is currently at the Indiana University, where he's been since 2018, and he directs a lab called Brain Networks and Behavior Lab. And they're focused on characterizing macro scale brain networks and looking at how that varies in related to cognition, disease, development, et cetera. So he's been doing a lot of interesting work, and we're looking forward to hearing what's going on with you in terms of your latest work today. So thanks again for joining us, and I'll pass it over to you now.
Excellent. Thank you so much for the introduction, Vince, and thank you, Armin, and everybody for the invitation to be here. I'm actually really excited to talk to specifically this group about some new work from my lab and from some of our close collaborators. The talk today is entitled Edge-centric connectomics, and I hope I'll convince you that there's something interesting there, and that you'll eventually see what that means over the course of the talk. So a brief outline, I will give a very short, very high-level introduction to network neuroscience, the space in which my lab works. I will then talk about this construct called Edge time series and how we can use this to measure time-varying cofluctuations. And I'll finish by talking about what we call edge-edge connectivity or edge functional connectivity, and how this can be used to probe the pervasively overlapping community structure in brain networks. So today will end up being it's a technical talk. It's about tools, but I'm gonna try to convince you that these tools are ultimately useful, maybe for your own work. So really a quick introduction. As I said, my lab starts with the premise that nervous systems are fundamentally complex networks. They're a special class of networks in the sense that they are multi-scale, meaning we can meaningfully talk about brain networks at different spatial scales, from the level of synaptic contacts between individual cells, individual neurons. Of course, C. elegans is the prime example of a cellular level connectome. The entire nervous system, chemical, electrical, and motor synapses have been mapped out in this micro scale connectome. We can also consider connectomes at the slightly coarser spatial scale of projections linking areas to one another. This is an example of a mouse connectome. These are axonal projections that span, that link different anatomically defined areas to one another. And all the way up to the macro scale, which as somebody who wants to understand human brains, we spend a lot of time at this spatial scale. This is an example of white matter tracts reconstructed from diffusion imaging and tractography, so delineating large-scale fiber bundles. Brain networks are also fundamentally multimodal, as you likely all know, which means we can talk about networks like I've shown you so far, which are all anatomical networks or structural networks. The connections in principle, you could crack open a skull and you could touch the white matter in principle. Don't do it. We can think of networks as being structural, representing these physical and material pathways between cells, areas, and regions. We can also consider functional brain networks, which measure something about the dynamic interactions between brain areas. So perhaps the canonical example for this group and for my work is the correlation structure of resting or task fMRI. So typically when we talk about functional connections or functional networks, we're talking about time series recorded from different points in the brain. They are functionally connected if they are usually correlated with one another, and otherwise we consider them not connected. So a prime example here is the seed positioned in posterior cingulate and medial prefrontal cortex, the blue and the orange curves, and you can track them across time. You'll notice that they follow very similar pathways. We would say that those areas are functionally connected with one another. A word on networks then. in general, these networks, irrespective of scale and modality, we can represent them as networks or graphs. The brain regions, the neural elements, the neuronal populations become our nodes, and the anatomical or functional connections become edges in this representation. It's a very simple model. It strips away a lot of biophysical detail and about nervous systems, but it's eminently simple. And its simplicity has allowed the network model to be applied across lots of disciplines, ranging from microbiology, where we can build networks of interacting with proteins, to medicine, where we can study diseases and disorders linked on the basis of their comorbidity with one another, to the migration and movement of cultural capital across time from one geospatial location to another, even to the environment in which organisms are embedded in. All of these systems, although very, very different, coming from very different fields, can all be represented meaningfully as networks. The network framework is common across these disciplines. It allows us to exchange computational tools. I can talk to a sociologist about community structure, and they can talk to me about their techniques for detecting communities and modules and networks. It allows for this sharing of ideas and tools. And to the extent that we care about it, many physicists are interested in, say, universal patterns, properties that are shared across all networks. So network science is a useful way of representing complex systems. Strips away lots of details and allows us to use the same framework in lots of different diverse contexts. When we build brain networks, however, we usually are considering two different flavors. The structural brain networks coming from tractography and functional brain networks coming from recordings of bold activity. In both cases, typically what happens is we chop our brain up into parcels or regions, and then for every pair of regions, we ask, are they connected by, say, a reconstructed streamline representing white matter, or is their activity statistically dependent on each other in some way? If so, we draw an edge between the two. These networks are represented as matrices, the sparse structural connectivity matrix, and a fully weighted and signed correlation matrix corresponding to functional connectivity. The way to read these matrices, if you're not familiar, they're essentially lookup tables of who is connected to who in the brain. Rows and columns correspond to brain regions or nodes in the network, and the entries indicate the presence and absence and weight of connections between pairs of regions. And if you follow like typical network analysis, or kind of like what people think of as like graph theory in neuroscience, these matrices then get subjected to all kinds of different measures that are purported to tell us something about properties of the network. For instance, a node's degree, the number of connections it makes, all things equal, tells us something about the capacity of that node to influence others in the network. We can study the extent to which systems are segregated from one another in the brain by tracking the modular structure, densely interconnected subgraphs, and even diagnosing hubs on the basis of how their connections are situated across modules. And we can also study how information and signals are integrated and flow across the network with measures based on, for instance, paths and walks. So network science is quite useful, and it's being applied fruitfully to brain networks. A few more words on functional and structural connectivity. I'm showing you two examples here, functional network on the left and a structural network on the right. At face value, you might notice some commonalities, and I'll toggle back and forth between the two to help convince you that there is some, albeit not one-to-one, but some relationship between the two. And indeed, these networks, these connectivity matrices are similar in some ways. Both structural and functional networks can be represented as matrices comprised of nodes and edges. They exhibit multi-scale topology, meaning that we can meaningfully query their structure at the scale of individual edges and nodes, all the way up to the network as a whole, and even at intermediate scales by studying its communities. But structural and functional connectivity are fundamentally different along lots of different dimensions. Structural connectivity is physical and material in nature. In principle, I will claim, at least over short time scales, the number of tracts, the number of white matter connections are finite, and we can map them in their entirety and completeness. White matter connections and structural connectivity have complex biophysical attributes, external density, strength, conduction velocities that are governed by biophysics. And structural connectivity does change, but it changes over long periods of time, generally speaking, of the course of development and aging, and with plasticity effects. Structural connectivity is this physical material backbone, and it shapes the interactions between regions, which means it constrains and shapes the flow of information or signaling that propagates over the network. Functional connectivity, on the other hand, is statistical in nature and dynamic. In principle, there's a large and almost infinite set of configurations that functional networks could adopt. And functional connections themselves have their own attributes, including persistence across time, directionality, and weights. But functional connectivity, because it is dynamic, it can change from moment to moment, and it is input and context dependent. So depending on the task an individual is performing, depending upon their current state, functional connectivity can change rapidly. And the way we think of functional connectivity, it is the outcome of all of the communication and signaling processes that are unfolding dynamically over the structural connectivity. And collectively, this comprises the modern field of connectomics or network neuroscience. And just one final word, this is a growing field. It has its own sub-disciplines now. Network neuroscience has its own subdomains, including studies that try to understand precisely what policies the brain uses to communicate, how functional networks and other networks change over short periods of time. We pulled tools from physics and network science proper to build so-called multi-layer network models. There's interest in how the brain's community structure, so dense subgraphs, change over short periods of time. There's interest in fingerprinting, so identifying connections that are, or regions whose connection profiles are distinguishable across individuals, and even kind of science fiction-y areas like network control. So how might we drive a system with exogenous input so as to push it into a particular brain state? And there's actually some nice empirical work suggesting that we can do these kinds of things. My lab works in this space, and this is the final kind of introduction few slides. But my lab works in this space, network neuroscience broadly. These are our papers from the lab. They're interlinked thematically. And share your code if you want to do this. It takes data from Google Scholar and builds these kinds of networks from your papers. But these are papers that are linked if they share common themes. And my work has lots of themes that, although it's diverse and sometimes applied, sometimes theoretical, it is interlinked by a small subset of themes. So my lab is interested in studying, say, mesoscale structure of networks, communities, and modules. We're interested in studying how network structure changes over time, both within a single scan session, but also across development and the lifespan. We are very interested in understanding the structural basis for functional connectivity, so studying structure-function relationships, and even the notion of wiring costs, so how brains negotiate the formation of costly, materially and metabolically costly connections, so that they can still nonetheless maximize their adaptability to the environment. But today, I'm going to tell you about what is for us a very new area, that is edge-centric connectomics. We started this just a couple of years ago, and it's been very fruitful for us and maybe controversial at times, but I want to tell you about this new area today. And a lot of the themes that I just talked about will appear at different points throughout the following slides. But this is, again, a relatively new area for my group and for my collaborators. Okay, so what I want to talk about first is I want to introduce our edge-centric framework through the construct of edge time series. Now, to understand this well, to understand what makes the edge-centric perspective useful, it's useful to contrast it with traditional, what we call traditional node-centric perspectives. So all brain networks, at least as we study them now, are composed of two elements. They must be composed of two elements. The nodes and vertices, which represent sensors or regions or areas, and then the functional or structural connections between those areas. I'm going to argue that network neuroscience, maybe even neuroscience proper, has been largely interested in properties of nodes, really since its inception as a field. This could be the number of connections that a node makes, its degree. the community assignments, the cluster to which a node is assigned, the centrality of a node with respect to some dynamical process unfolding over the network. This node-centric perspective is pervasive across the discipline and it really parallels a rich history of parceling, mapping, and labeling areas in neuroscience, going at least back to Brodmann, but also there's evidence of this in more recent efforts to, say, parcelate brains on the basis of anatomy or jointly anatomy and function. But again, always the focus has historically been, I would argue, on nodes and areas and populations. This doesn't mean, however, that there are no efforts to prioritize edges over nodes. And there's some very, very nice examples of this coming from other disciplines, from network science proper. I won't dwell on this, but I want to use this just to kind of motivate our work. So some examples of frameworks that prioritize edges over nodes are, say, link similarity. This is 2010 Nature paper from YY An and Lajla Barabashi. And essentially what this approach does is it transforms a traditional node-node connectivity matrix, a node-node network, into an edge-edge network, where the edges become the units of interest. And it does it in a really simple way, I think elegant way. It considers two edges, and these two edges must have a shared stub, a stub node. So here they share this node, which leaves two unpaired stubs. And what YY's group did was they just calculated how much overlap there was in the connectivity profiles of those two unmatched stubs. In this case, there's 3 total connections. They have 1 shared connection. So the partial overlap is 1 out of 3. It's a Jacquard index. If you do this for all pairs of edges, what you end up with is an edge-edge network. So now the rows and columns don't correspond to nodes in this connectivity matrix, but to edges. The advantage, or at least one of the interesting advantages of this approach, is that when you say cluster an edge network, you're assigning the connections to communities, you're assigning the connections to modules, so that from the perspective of a given node, it maintains affiliations with multiple communities. The communities of its edges determine its participation in any one of these red, green, or blue communities. And of course, there's other approaches to, say, the line graph approach, which transforms a network first into its incidence matrix, and then takes the outer product of the matrix with itself to generate another edge-edge network. You can see these are for the same network, and the link similarity and line graph broadly have some shared commonality. And so these are 2 approaches. They both transform node-node networks into edge-edge networks, which is kind of nice, but there's some caveats. If we wanted to apply this to brain imaging data, say functional connectivity, it's actually not straightforward to do. These 2 approaches are best suited for networks that are sparse, as opposed to fully weighted and signed networks like functional connectivity measured with correlation. And these approaches don't really have a convenient way for handling signed connections. So it's common, even without global signal regression, to discover some small proportion of negative weights in a functional network. These 2 approaches, if we wanted to build an edge-edge network from those functional connectivity data, it doesn't really have a way of distinguishing between positive and negative connections. But also, and for us this is really important, these 2 approaches, their transformation of the networks, of the graphs themselves, which have no temporal properties. So if we were to apply these to functional connectivity, we lose all access to time varying properties, to the time series that generated the graphs. And selfishly, I want that kind of information in my analyses. So back in 2019, these are pre-pandemic days, we started asking ourselves, if we wanted to take an edge-centric approach to brain imaging or brain network data, what would we do? How would we do this? And we spent a lot of time thinking about this. This, by the way, is work that comes from, it's a big collaboration with lots of people, but led by my postdoc, Farnaz Zamani, and ultimately Josh Faskowitz too. We started asking ourselves, what would an edge-centric approach look like? And eventually we settled on one particular framework. And it starts with our traditional way of defining a functional connection. So let me walk you through this. So suppose we take activity from 2 different parts of the brain. If we wanted to measure the functional connectivity weight between those two areas. The most common approach is to use the bivariate, I hate using the word Pearson, but bivariate correlation between those two areas. We literally ask, are they correlated with one another? That gives us a correlation. That becomes the connection weight in the functional connectivity matrix. We do this for all pairs of brain regions, and it fills out that matrix for us. I want to unpack this step though, and bear with me. I'm not trying to insult your intelligence, but I want to walk through how we calculate a correlation. So the way we calculate a correlation is like this. We take our raw, untransformed time series, z-score it, subtract the mean, divide by the variance. To calculate the correlation, we then multiply every single frame against itself in the other time series. So we're multiplying at a frame-wise level the z-scored activity. Do this for all frames, sum, normalize, and that gives us our correlation coefficient. That is the simplest way you can get a correlation. But what if we skip that last step? What if we skip that summation step? What do we get? Well, first off, we lose our correlation. We don't have this time averaged cofluctuation, the mean cofluctuation. Instead, we have a time series. We get the product of these z-scored time series at each instant. We're going to call that a cofluctuation or edge time series. The elements in this time series, they tell us when the activity of two regions are deflecting above or below their mean and by how much. And I'll give you a couple examples. Here's an instant when the cofluctuation time series has a big positive value. Why is it big and why is it positive? Well, because at that instant in time, the red and blue time series are both deflecting in the same direction and by a pretty sizable amount. So when we multiply the two together, we get a big positive value. It indicates that those two regions are deflecting in the same direction and by a big amount. We see an even bigger negative deflection in the cofluctuation time series at an earlier point. And this is because the red time series is at a very large value above its mean, and blue below its mean. And we get this big negative cofluctuation. And we get one of these cofluctuation time series for every pair of brain regions. What I'm going to claim, we can debate this, I'm happy to, is that these cofluctuation or edge time series have interesting, so just like intellectually interesting properties, but also I'm going to claim useful properties for neuroscientists and cognitive neuroscientists and network neuroscientists. And I'm going to tell you about that in the following slides. Okay, so let's say we repeat this procedure now for all pairs of nodes, and what we get back is a matrix that looks something like this: time by edges. If we were to slice this across, for any given edge, it gives us a single cofluctuation time series. If we were to slice this at any instant in time, we have the instantaneous cofluctuation between all pairs of brain regions. These are the upper triangle elements of a node-by-node matrix, which we can essentially regard as a network. It is giving us instantaneous time-varying cofluctuations. I actually regret using the word FC here because we're not measuring a correlation. We're measuring instantaneous cofluctuations. We can still treat it and model it as a network, but I have no expectations that this should behave like a correlation. It is unbounded in principle. It is not the same as time-varying connectivity with a sliding window and correlations. I want to make that point. So it gives us these time-varying properties. But I will point out, though, when we do compare this to sliding windows, we don't have to use a sliding window with the cofluctuation time series. There's no blurring, and as a consequence, we can resolve with frame-wise resolution any kind of cofluctuation in the network. So we get very bursty, high-frequency kind of changes, whereas with even very, very short sliding windows. In this case, only 10 frames, so like 7 seconds, which is far too short to, I think, that most people would claim is useful. We end up blurring a lot. We lose some of this rich temporal information. So there is some potential advantages to using this edge time series or cofluctuation time series approach. It also has other advantages too. The average of a single cofluctuation time series is a correlation coefficient, which means if we average every edge's edge time series, we can rearrange those elements into the upper triangle of a node-by-node or region-by-region matrix, and it returns exactly functional connectivity as measured with a correlation. So co-fluctuation time series are a mathematically precise decomposition of your functional connectivity. As long as you're measuring it as a correlation, it is a precise decomposition. You add everything up and returns exactly your correlation weight. And it is also giving us frame-wise estimates of coactivity or co-fluctuations, which is something that is amenable then to a network analysis. So we started doing this. We started applying this approach to some data. And what we noticed is that when we looked at these edge time series, it looked like it kind of had bursty behavior, where we saw some really high amplitude frames punctuated by long periods of quiescence. And we started asking ourselves then, well, do these high amplitude frames, do they tend to occur simultaneously across edges, or are they occurring independently? Right? Do we see something that looks like this, where we have different edge or co-fluctuation time series? Those bursty frames are more or less aligned with one another. In which case, because the co-fluctuation time series are an average, their average is functional connectivity. It would mean that those frames then might be more informative because they have larger values, they contribute more to that average. Or are these high amplitude instances, are they more or less independent and spread out and smeared out across time, not occurring coincidentally? So if you have good eyes and you're following very closely, you might notice these striations in this matrix, in our edge time series matrix, in which case you already know the answer to that question. These striations indicate instants in time where lots of node pairs, many edges, simultaneously express very high amplitude co-fluctuations. We can summarize these co-fluctuation amplitudes by calculating the root sum of squares or root mean of squares across all edges. And we find that this distribution is more or less heavy-tailed, so there are many frames with small amplitude, but a very small number with extremely high amplitude. We have, because we're not very good with naming things, we call these events. Apologies to the EEG literature, where events have a specific meaning, but we're calling these high amplitude frames events. And just to indicate what how distinctive these frames are, these are the highest amplitude frames in this edge time series. These are the lowest. So at the low, it's very difficult to even find any kind of co-fluctuation, but at the highest amplitude, there's evidence of lots of edges simultaneously exhibiting very strong positive or negative co-fluctuations. And to us, this suggested then that at a frame-wise resolution, as measured with co-fluctuation time series, our network activity is bursty. It's transitioning between these periods of high amplitude co-fluctuations and then periods of quiescence. This is not a new idea, I should point out. There's lots of work that has hinted at similar phenomena in fMRI and other modalities. But I will point out that our approach, which I think makes it distinct from these others, is a precise decomposition mathematically. It is parameter free. There's no threshold for an event. There's no, we don't have to specify a seed region, for instance. So it gives us a little bit of, I think, above and beyond what some of these other very nice papers have contributed. Yeah, so what are the implications for this then? Well, it becomes clear to us that when we compare the behavior of the tails of this RSS distribution. So on the left-hand side here, this is the functional connectivity reconstructed using only the highest amplitude frames, the top 5%. And on the right is the bottom 5%. And we asked, well, how similar is this pattern on the left to functional connectivity, the static functional connectivity, and then do the same for the pattern on the left. And we observed that even 5% of the frames, a very small number, the high amplitude frames were highly correlated with static functional connectivity, whereas the low amplitude frames, not that they weren't, but they explained much less variance, suggesting to us that these high amplitude frames contribute disproportionately in shaping that static functional connectivity. And in some ways it must be, because remember, correlations are the sum of co-fluctuation time series. So if there are frames that have very high amplitude co-fluctuations, they necessarily contribute more. Relatedly, we find that those high amplitude frames are also highly modular. so that there are communities expressed during those frames that are highly segregated, much more so than those bottom low amplitude frames. I'm showing you examples here, by the way, with 5% of the top and bottom, but this effect holds literally for every percentile, whether we're looking at just the top 1% versus the bottom 1% and so on. It holds at literally every level. So this, to us, our interpretation was that functional connectivity, then static functional connectivity, is measured as a bivariate correlation, and the systems that are expressed in those connectivity matrices, they can be partially explained or attributed to these infrequent, brain-wide, high amplitude co-fluctuations, which was very interesting to us, and at the time, kind of surprising. And since this first paper, we followed it up with a bunch of I'm not going to say lesser papers, but more nuanced papers investigating more specific questions. So what else have we seen? We've looked at what the activity patterns look like during events, during these high amplitude periods. And they're largely underpinned by a single mode of brain activity, one that emphasizes, depending on who you talk to, maybe an intrinsic versus extrinsic networks. The first principal gradient, if you talk to gradients people, looks like a DMN versus kind of everything else, but it's a very recognizable pattern of activity. We have found that the events, the amplitude synchronizes during movie watching. So the events occur synchronously across individuals, and I'll have more to say about that in a moment. And we've also found that these event periods, these brief periods, they actually contain very strong individualized information about subjects, as well as strengthened brain behavior correlations. And we show this with Midnight Scan Club data for the identifiability part, where we reconstruct networks from the highest and lowest amplitude frames. And we use this identifiability measure to show that during the high amplitude frames, the networks that we reconstruct are highly similar within subjects, but dissimilar between. And the opposite is true during the lowest amplitude frames. The lowest amplitude frames contain very little subject-specific information. And again, with HCP data now, we again reconstructed networks for the highest and lowest amplitude frames and use those to predict principal components derived from behavioral and demographic measures. And this is the first 10 components that we derived. And on the left-hand side, you see the correlations that we obtained using the high amplitude frames, the top versus the low amplitude frames. In every case, the correlation magnitude exceeded that of the low amplitude. So the high amplitude frames contain some subject-specific and behaviorally relevant information that's not available in the lower amplitude frames. Just a few more notes on follow-up studies. After this first paper, we actually went back and clustered events, and we found that they are not, in fact, monolithic. There are different flavors of events, different flavors of co-fluctuation patterns. My undergraduate, Sarah Greenwell, using 28andMe data collected from Emily Jacobs' lab at UCSB. She found that particular patterns of co-fluctuations, particular states, are associated with endogenous fluctuations in sex hormones within an individual across 2 menstrual cycles. We have established, this is work from Maria Pope in Olaf Sporn's lab, we have established a structural basis for events. So in her 2021 paper, she used a simple oscillatory model to generate synthetic time series that are structurally constrained using white matter connectivity. And she found that with the intact network, we found events within the synthetic data, but when we destroyed the modular structure of the network, the events went away, suggesting that the underlying structural connectivity has a key role to play in shaping the emergence of events, and specifically its modular structure. And more recently, a graduate student in my lab, Jacob Tanner, has investigated the movie watching data in further detail. And what he found was kind of interesting to us. He found that, indeed, as we saw in our previous study, events did synchronize across individuals, but specifically where these red lines are, these are boundaries between movie clips. So these are short vignettes, 2 or 3 minutes long. And almost everybody, right at those boundaries, have events. Every row is a subject, and so this is looking at the events across time. So events tend to occur immediately following the ending of movie scenes. We kind of don't know what to make of it. We have some hypotheses about what this might be, some mostly speculative at this point. but it was quite interesting to us and striking. We see synchronization within the movies too, but specifically at these boundaries, we see synchronous events occurring across almost every subject, which was again quite interesting to us. Okay, so that was our kind of Edge time series story. I have just a little bit more to tell you about. And this is now moving into what I'm going to call edge connectivity, or edge functional connectivity. So I've been talking a lot now about these Edge time series. Again, my group is very excited about them. They tell us something about how edge weights vary across time, and it's a mathematical decomposition, and we're quite fond of them. We started asking ourselves right around the same time that Farnaz's PNAS paper came out, like what else can we do with Edge time series? And I see a question from the audience. Yes, the movie data is available. This is actually HCP 17 movie watching data. We've, by the way, replicated all of this with in-house data collected here at Indiana, which is also available in the supplement of Farnaz's PNAS paper. There's a link to a Figshare repository with all of the data. Anyway, we started asking what else can we do with co-fluctuation time series. We said, well, if we can calculate correlations between regional bold, why can't we calculate correlations between edge level co-fluctuations? So we did. We calculate the similarity between every edge or co-fluctuation time series. This gives us a huge matrix, by the way. It's a node pair by node pair matrix, edge by edge. So our original study had 200 nodes. This is using Alex Schaefer's 200 node parcellation, which means we have roughly 20,000 unique edges in our network. And so our edge matrix, the weights which we're calling edge functional connectivity, because again, we're not really good at coming up with original names for things, but it's a massive matrix, fully weighted, double precision, similarity values between every pair of edges. This is a networky science talk, so I have to show at least one embedding diagram. This is edge functional connectivity, EFC, with a spring embedding. So here, when we're used to seeing these, normally the points, the vertices represent brain regions or nodes. Here, the vertices represent edges. They represent pairs of brain regions, and they're linked to one another if their edge co-fluctuation time series are highly similar. So this is, of course, thresholded for the sake of visualization, but in reality, weights exist between all pairs of edges, even if they're very, very weak. So what do we do in this paper? Well, we do a lot. With Scan Club data, we show that edge functional connectivity is highly repeatable within individuals, as long as you have enough samples. My graduate student, Younghan Jo, showed that indeed there is a high level of subject specificity, and that the fingerprint that we could derive from edge functional connectivity, it actually outperforms the fingerprints that we derived from nodal functional connectivity, suggesting that something about this higher order edge level representation of a brain network, it reinforces somehow subject level traits, it locks in subject level connectivity in a way that node connectivity does not. We also showed that edge functional connectivity is subtly but systematically modulated by brain state. So we compared rest with movie watching and found found differences. In principle, if you were so inclined, you could carry out a network analysis, a graph theoretic analysis on the edge matrix. In which case, you calculate a degree, for instance, you'd be getting a degree per edge. You could then embed that in a node by node matrix. So every edge gets its degree. So we haven't followed this up very much, but in principle, you could carry out network analyses on the edge connectivity matrix as well. We've also looked since then at motifs, which is another form of graph analysis or network analysis. In the paper, we examine brain behavior correlations and do a lot of benchmarking to confirm very obvious confounds like respiration and cardiac artifacts and emotion, of course, which doesn't seem to be impacting the events or the edge functional connectivity very much. But what I think is especially useful about the edge connectivity, what I find especially compelling, is its implications for system level organization in the brain. So I'm going to show you some diagrams really quick, some figures from other people's papers. I think probably most people here recognize this right away. This is from Thomas Yao's seminal 2011 paper, in which he clustered a bunch of vertices in the brain on the basis of their functional connectivity similarity. In this paper, they derived, depending on who you talk to, 7 or 17 clusters. These clusters are so ingrained in cogneuro and human imaging now. They have names. We've given them names. There's a purple visual cluster, limbic, default mode network, an attention, motor, control, another attention network. These are names that we've ascribed to them that are functionally evocative, by the way. They tell us something supposedly about what these systems do functionally. If you don't like Thomas's work, maybe you like John Power's work, which came out more or less contemporaneous with Thomas's. Again, clustering functional connectivity on the basis of similarity and connectivity profiles. And more or less, there's pretty good concordance between the two. A lot of the same systems are detected. Thomas's paper had one motor network. John Power's paper had two. But again, a high level of similarity. More recently, Evan Gordon's paper. If you don't like either of these, you can use the Gordon Atlas. If you don't like that, you can use the Shen Atlas from the Yale group. All of these divide cortical territories into putative brain systems. And they all do it in a very specific way. Namely, they force the system labels to be non-overlapping. Surface verdicts, a parcel, they belong to one system and one system only. I'm going to argue that a lot of this is a methodological convenience. There's no reason why somebody couldn't apply a method that allows for a little bit of overlap, but it also disagrees with what we know about neuroscience. There have been countless studies that have recorded electrophysiological and calcium data from very specific functional territories, say visual cortex, that we think of as being functionally unimodal, but have later found out that some of the variants, significant fractions of the variants in cells' responses, are attributed to non-visual stimuli, for example, in mice, like whisking behavior and movement in their environments. So all these systems that are one-to-one mappings of vertices to system label, it's useful. It's a framework that my lab still uses a lot, but it is a kind of a convenience that I think is largely bound to as a methodological convenience. So how can we inform this debate? How can we contribute here? Well, we can cluster edge functional connectivity. And I'm going to show you some examples of this. So here are edges, the edge time series reordered by cluster. Here's the edge functional connectivity matrix, again, reordered by, chose 10 clusters here. It's not necessarily representative, but by 10 different clusters. And here's the co-assignment probabilities across repeated runs of the clustering algorithm. But remember, we are now clustering edges. We're not clustering nodes, which means that from the perspective of a given node, it actually can maintain multiple affiliations. Its edges align it with different clusters, potentially different, not necessarily a single cluster, but very likely different cluster labels. So whereas those slides I showed you, or the figures I showed you on the previous slide, the clusters had no overlap, could in principle relax that. So there's a little bit of overlap, and people have. But our approach I'm going to argue, exhibits what we call pervasive overlap, because we're clustering edges and nodes are linked to different communities on the basis of their edge cluster assignments. We end up with from a node level perspective, a cluster profile that is fully overlapping. Every node participates in some, or in a plurality of clusters. And here I'm showing you, this is from the paper, the edge clusters mapped back into a node by node matrix. So the colors here indicate the cluster to which an edge is assigned. I can show you some representative co-fluctuation time series from within each cluster. Here, for example, a cluster of edges that include lots of edges that fall within somatomotor networks, but also edges that link somatomotor with salient ventral attention networks and dorsal attention networks. All of these edges behave very similarly to one another. The connections exhibit similar properties across time. Same is true with this visual and dorsal attention block, this more complicated configuration, but they all exhibit cohesive co-fluctuations across time. This is the key point here. We can, of course, project these edge level assignments back to individual regions, literally by masking this matrix for each community and averaging across its rows. Occasionally we find communities that resemble known resting state networks. For example, this is community 2. As you see, most of the edges are incident upon somatomotor network. So the resulting pattern looks a lot like a motor cluster or a motor ICA map. But there's other communities, other edge communities that we projected back to individual nodes. It has a resemblance to something familiar, like maybe a control or a default node network, but it pulls along other systems with it. So here's an example of something that's largely maybe DMN or control network-esque, but it has some contribution from edges in the somatomotor network and maybe some salience nodes as well. We can use these projections and actually calculate where the highest level of overlap is in the brain. Now, if you were to ask me where this would be before we did this study, I would probably tell you association cortices, areas associated with control networks or something like that, or frontal parietal networks. Somewhat surprisingly, we found the exact opposite. The highest level of overlap, the regions that participated in the most diverse set of edge communities, were actually located in primary sensory and attentional networks. So visual networks, motor networks, salience, ventral attention, and dorsal attention networks. Control and default mode, those tended to have actually the lowest levels of overlap. This was kind of perplexing to us because it really runs contrary to almost every study of nodal functional connectivity. So John Power has done a lot of really great work, followed up his 2011 paper with this 2013 paper. And in it, he looked at a measure called participation coefficient. So the extent to which regions participate in different non-overlapping communities. Well, they found among the highest level of overlap was in control networks. We did not find that. Max Bertolero, using a similar measure but applied to a very different data set, found basically the same thing. Thomas Yao found the same thing. The color map here should be inverted, really. The green cells, the green areas actually indicate high levels of overlap, whereas the red indicate low. But again, they found something very different to what we found. So we start asking ourselves, well, where the heck did this overlap go? Why do we see no overlap in our, or no functional plurality in control networks? And so to address this, we started poking around a little bit, and we discovered something that I think is quite interesting. So what we started looking at was what I'm going to call edge community profiles. So for every region, we take that edge community matrix as the community labels projected onto edges, and we take slices of that corresponding to individual brain regions. And we can ask, how similar are these two regions in terms of their edges community assignments? We can do this for all pairs of regions. And we get a map that looks something like this, an edge cluster similarity. And what we found was that when we looked at the within system similarity, so how homogeneous these profiles are with respect to one another, the control networks are incredibly diverse and internally heterogeneous, which means that the nodes in those systems, on the basis of their edge community profiles, are all very, very different from one another. As a system, they're internally diverse. In contrast, primary sensory systems like the motor and visual networks, they're much more homogeneous internally. All of the regions within those systems tend to have similar edge community profiles. So this suggested to us, or suggested the possibility that maybe some of the functional diversity in heteromodal systems like the control network, it's not really a property of nodes or edges per se, but it is really expressed at the level of the system. In order to appreciate the diversity and the heterogeneity, we need to examine the system as a whole. It's really quick. I will summarize really quick. I think I have one more slide after this, but if we put this all together, what have we learned? Well, we show that this co-fluctuation or edge time series are an exact decomposition of functional connectivity, and allows us to track moment-to-moment changes in the co-fluctuations at a frame-wise temporal resolution. Our analysis of this has revealed so-called bursty dynamics, these events followed by periods of relative quiescence. And I'm reluctant to use the word drive, but I've included it here. But the events contribute to disproportionately to the organization of static functional connectivity and its system level architecture. And there's some hints that this might be a useful approach for studying brain behavior relationships in the future by selectively filtering time points and reconstructing functional connectivity from select frames. In Josh's Nature Neuroscience paper, in turn, we found that we can actually take those edge time series one step further by calculating edge functional connectivity, and it reveals these cohesive sets of edges that are co-fluctuating in similar ways. And when we cluster these edges, they give us pervasively, fundamentally overlapping communities with marked heterogeneity across brain systems. So I have one final summary slide, but just like and a kind of an opinion or perspective on all of this. And I want to be very clear about this. I have no stake in this, truly, but I don't want to come across as saying that like edge-centric approaches are better than anything that's out there. I don't believe that, actually. What I do believe is that the conclusions we derive about nervous systems and brains from imaging data really depends on our choice of how we analyze those systems. At the end of the day, all of us here probably are studying a brain, human brain, a monkey brain. We're studying a nervous system. Sometimes we can learn a lot about this system by doing relatively simple things like studying activation patterns. We can learn a lot without ever talking about networks, I would claim. But occasionally we can learn something additional by shining a network perspective at it and looking at the shadows that emerge. And my hope, and this is kind of how I see the edge-centric approach, it's going to create its own shadow. When we analyze a brain with the edge-centric perspective, it might have some unique contributions as well. I'm not claiming that those supersede network-based approaches or activation-based approaches or ICA-based approaches, you name it, only that it is a complementary tool that occasionally can, I think, contribute some unique insight. And so with that, I'm going to stop. I just want to thank everybody involved in this. This is a small subset of the papers. These are, again, a small subset of the people who have been involved in this process. And by the way, if you're really tickled by Edge Time series, we do have still a position open. It'll probably be open for 2 years, assuming we get our NCE, to continue doing this kind of work. And all of our code and data is available on the lab website. You can always harass me on Twitter if you like. I'll stop here.
Thanks, Rick. That was great. I'm disappointed you said at the end that you're not claiming your method's better than anything else. I was hoping we could all attack you and have this.
You can still do that. Yeah. I'm trying to be diplomatic these days.
Very good, very good. Yeah. Yeah, the best hammer is the one that you have in your hand, right? So
hopefully not to hit me with, but yeah.
Right. That's why virtual meetings are useful sometimes.
That's right. I'm gonna stick to these for now. Yeah.
So if folks have questions, please send it to the chat. We can also unmute you and let you ask questions yourself. I guess I will start with one sort of question about the setup that you started with, which is when you were kind of talking about structural and functional information. And I think your sort of claim was that the structural information is sort of physical material, whereas the functional is statistical, right? And I guess the question I have is that really true at the macro scale? And I don't know what your thoughts are about that, because I see a lot of papers that sort of take structure as fixed and determined and then drive everything else from that. And I wonder if that's actually causing some missing some things, right, in terms of the strengths of the assumptions. So
yeah, I think at like as a 0th order approximation, like I do think that that is my perspective. Like structural connectivity is over short time scales, relatively immutable. It is finite, and something about structure, it constrains interactions between brain regions.
Yeah,
it shapes.
Well, I agree with that, but I guess what I'm talking about is the measurement that we've done. Like, so with an MRI scan, your voxels are huge, right? And so is that sort of observation that we have also consistent with that assumption?
I agree with you that, especially with tractography and all the limitations
Yeah.
associated with that, there are huge swaths and known tracts that we are biased against missing. So I think that is, in human data, a very real problem. We take structural connectivity as the best we can get non-invasively, but it's absolutely missing connections, and those definitely have impact on our models. I tend to liken like the current state of structural connectivity. It's kind of like the early sailing maps, where like they have Florida in the right place. They know there's a big bay somewhere near Virginia, but don't believe the inlets, kind of thing. So I think it locks in a lot of the large scale tracks that we do expect. But all the details I have very little faith in personally.
Yeah.
I have a little bit more faith that this general like way of thinking about structure shaping function, I have a little bit of faith that it is not wholly incorrect on the basis of reproducibility with non-human data. So people have done very similar studies with tract tracing in macaque and mouse, and used those so-called gold standard tracts, gold standard structural connectivity to constrain similar like neural mass models. And a lot of the same kinds, like the same flavor of results come out of that. But I take the point that it all depends on the quality and the accuracy of the input data. And I don't know to what extent that propagates to the level of the kind of conclusions we make. There's actually a really nice paper from maybe 2013. I think from Victor Yersa and Gustavo Deca. I think it came out in Journal of Neuroscience, where they played the game like, what if we started adding synthetically connections back to our structural connectivity matrix? Can we improve the correspondence with the synthetic functional connectivity to real functional connectivity? And it turns out when they place connections in the areas, between the areas that we know we were biased to miss, maybe not surprisingly, but the correspondence improves pretty dramatically. So definitely missing stuff. But I take the point. It's still kind of the Wild West, at least from my perspective.
Well, yeah. I think a lot of the work that we tend to focus on is sort of more on the sort of probabilistic parcel side, right? Data-driven parcels and more of a symmetric structure function as opposed to one way.
Yeah.
Kind of, and I think you had one slide that did have a bi-directional.
And definitely, I think over longer time scales, function definitely feeds back to shape structure. I think these are the kind of plasticity effects. I think even on this slide, I talk about a little bit, and certainly over early development, that plays a role in shaping the organization of this network.
Yeah, I mean, I'm not even talking about changes over time. I'm more talking about discovering the underlying structure of the representation of the brain functionally and structurally, right? So it's more, as opposed to just using an atlas, for example. So yeah, but anyway, let me have I've got a long list of questions, but I don't want to dominate the thing. So a question from Lisa was, how do you deal with respiration and heart rate in terms of the events that you're studying?
We followed, well, I can't remember whose paper it was, maybe a paper from Molly Bright or somebody like that. It's in the supplement of the PNAS paper. This is something that Farnaz did. Offhand, I can't recall, but we followed what we thought at the time was the best approach.
Do you collect heart rate and respiration data?
This was from HCP data, so that was included in the physio measurements. So we had those, and we did not have it with the Indiana data. I think we didn't have one or both of those. We had motion, and it behaved very similarly to the HCP data. But with HCP, and since then we have gone back and checked with other data sets and do have these other measurements, and so far those don't have a clear correspondence with the timing of events. I regard it still as an open question. We are not a physiology lab. We're not that's certainly not our strength. But I won't say who, but a lab that is very good at that had reached out to us at one point, and they purportedly agreed with us too. That it's not clearly they're like, wow, I'm surprised it doesn't align with tidal flows or something like that, or some physiological measurement or CO2. So I have a little bit of faith in that. Yeah.
Yeah.
It's also in the synthetic data too, where there is no
Yeah, yeah. I'm going to ask one more question that Armin may want to pipe in on this too, but you're talking about sort of the tails of the distribution, in terms of high versus low and disproportionate contributions for the high, which, I think is true. But if you look at the data in terms of, again, like parcels, sort of data-driven parcels and overlap and things like that, there's a lot going on at the low end that gets sort of missed. And so it kind of gets at your sort of focus on overlap, right? So interactions, I think that kind of is in the same spirit of that. But like Armin had done some work, looking at like these the CAP studies, for example, and showed, for example, that if you look at the lowest amplitude information, you see a lot of really interesting patterns kind of coming in terms of overlap between networks that are missed if you just focus on everything's got a purpose, right? You can look at it in different ways, but there are trade-offs there. So I don't know if Armin wanted to add anything to that. Yeah.
I totally agree with the statement, and Armin, I'm eager to hear what you have to say. I like that paper very much, by the way. I also don't mean to misrepresent our viewpoint. Initially, we did start off in this dichotomous low versus high. We have since relaxed this a little bit, and there's a couple of papers now, still I think in preprint stage, that have looked at those, maybe not the lowest, but those intermediate amplitude co-fluctuations. And what we discovered is a lot of the high amplitude co-fluctuations, they really reflect like when big networks come online, and those middle frames, or those middle amplitude frames, are the relatively smaller networks kind of popping up here and there too. So there's definitely structure in there. I don't want to give the impression that there's nothing happening in the middle amplitude frames. There's definitely interesting things happening. We did also, just one other really quick point. Now in multiple data sets, we have found at the very, very lowest amplitude frames, those are the ones that are almost always more strongly associated with in-scanner motion, like DVAR, like high DVARs values and high frame-wise displacements. The very, very lowest like bins that we've looked at. So we've informally even called them things like the garbage bin and kind of things like that, just because it's been so overwhelmingly motion contaminated. I don't know that this extends to every way of measuring amplitude, but at least
I don't think. I guess I'm missing that's maybe a little bit different than what we were doing, because we did look at the low amplitude relationship to motion. Armin, didn't you?
Yes, we did. In fact, I'm kind of confused because usually we see these heavy motion, like a fast motion in the low amplitude as well, right? So I'm not quite sure what Rick is referring to, because we look at also motion, we see like all those network, even after regressing out all those different first order, second order motion, we can capture those networks. So I'm not quite sure.
Yeah, it was. I guess ours was far from garbage when we looked at it. So I guess it seems to be. Maybe we're probably talking about apples and oranges. We'll have to look.
I think there might be a little bit of that. Yeah.
Okay. Well, there was a question from Cyrus about. I know I think I know your answer to this, but is the response from a repeated view of a movie exactly the same as the first response?
Starting with the idea that brains and R&D events at the end of each are just the standard.
Is the response from a repeated view of a movie? I think he's just saying, is there a history effect? When you look at. Yeah.
No. So maybe I'm misunderstanding the question, but I don't think that they're all the same. The ending response. I don't think they're all identical, but I do think the nature of the high amplitude co-fluctuations, they are repeatable. So the HCP17 dataset has one really unique feature, and that's that at the end of every movie scan, they show the same clips again. So they have 4 scans of the same individuals, but at the very end, they have repeated. You see the same stimuli multiple times. We see no attenuation in the amplitudes after repeated viewings, but we do find that the events, they all tend to be very high amplitude, shared across individuals, and the timing is almost identical across these different watchings of the same movie. But maybe I'm misunderstanding the question.
Yeah. No, I think, yeah. I think that's that's helpful. I wanted to ask, I guess I have to bring, it's 12:05, but I'll ask one more question from Ashkan, which is
Whew, so long.
he's got a couple long questions. I don't know if, they're very interesting.
Yep.
We should definitely have it, but sort of the question of node. You claim that the focus was on nodes, and he is asking if, well, in actuality, if the nodes are essentially some type of networks themselves. So like, I think that's kind of the similar question. It's related to the question I asked about overlap, and probabilistic parcellations that overlap, right?
Yes.
And then working on them as edges is kind of working on the edges of the original data the nodes were estimated from. So a lot of work has been working on edges, even though they haven't been named edges in a sense. So maybe it's not that the work out there is node focused. It's just that it hasn't been, it's been all labeled as nodes, but maybe it's not, right?
I have to admit, I don't fully follow the logic of the question. I'm trying to read it carefully.
Yeah.
Ashkan, I think we talked maybe at an OHBM or two ago about some of this, but I'd be happy to follow up online too.
Yeah.
I have a slightly.
There's going to be a Slack. There's going to be a Slack follow-up, and Armin will save these questions, and I think we should continue there. This is probably a more detailed and pretty fun, probably, conversation. So everybody, stay tuned. We'll have continued, interactions with Rick in the Slack channel. So I guess in the interest of time, I'm sorry we kind of have so many. There's a lot of interest here. This is great. So the talk is recorded. The slides, and information will be available via the Slack channel and other means that will be emailed out. And probably we should just close the session and thank you, Rick, for joining us. Yeah, it's kind of like we almost we almost want to have like a whole hour for discussion after each talk, right? Because we could keep we'll have you out here at some point. We can go grab some coffee. It would be fun. So
Great.
But in the virtual world, we'll work in Slack going forward. So so anyway, sorry to kind of rush a little bit at the end. Armin, any other comments?
No, just want to make sure, Rick, you already joined the Slack, right? So if
I got the invitation, I think I even signed in. Yeah.
great, great. I apologize for everyone who has a question, raise a hand. We will continue on the Slack. So thank you again. Thank you very much.
Thank you for the invitation.
Yeah.
This is fantastic.
Have a great weekend, everybody.
See you, everybody.
Okay. Thanks again, Rick. Okay. Bye-bye.
Bye-bye.